Nice to see everybody. You've got the exam coming up today during the laboratory period, so hopefully you're all studied up for that. I'm going to continue this lecture on signal transduction, which remember is not going to be on this midterm, it'll be on the next midterm. And probably we'll finish up signal transduction lecture today and then go on with the next topic, which is chromosomes. All right, well, let's see. Let me restart with the G proteins. Or maybe I should recap here that the whole idea is that cells sometimes need to communicate with other cells, and the cell that is sending out the signal releases a molecule called a signal molecule. And the cell that's going to receive the signal, which will be this big thing right here, has to have a receptor to detect that signal molecule. And one of the points from last week was that there's three major types of signal transduction pathways that these cells have, three big families of signal transduction pathways. And the signal transduction pathway is going from binding the signal molecule with a receptor to activating what's called the cellular response enzyme. And anyway, there are three major families of these receptors that start the signal transduction process. We talked about tyrosine kinase family of signal molecule receptors. And we finished off last time talking about the G protein linked receptor family of signal molecule receptors. And let's review that one. In these G protein linked receptors, there's a receptor for the signal molecule. And what's kind of interesting is, in these G protein linked receptors, it's a receptor molecule, a protein, but it has 7 hydrophobic transmembrane domains. For some reason it crisscrosses through the membrane 7 times, and I don't know why that is. I never heard anybody offer a reason why these family of receptors have that particular structure, but they do. And when they bind the signal molecule, I'll use that as a signal molecule shape. When they bind the signal molecule, remember that what happens is when a receptor binds a signal molecule, it has to activate what are called the relay proteins. And the relay proteins are proteins that eventually, one activates another, activates another, activates another, until you eventually get down to activating the cellular response enzyme. Well, the first relay protein, the one that's activated by the G protein linked receptor, is called a G protein. And these G proteins are membrane proteins. They also have hydrophobic domains that keep them anchored to the membrane. Does anybody recall why they call these G proteins? It was something that they bind onto. A G nucleotide, RNA nucleotide. Before these G proteins are activated, in other words, before the receptor binds its signal molecule, the G protein is kept in an inactivated state because it's binding a guanosine diphosphate RNA nucleotide. And for some reason, that RNA nucleotide acts as an inhibitor, an allosteric inhibitor of the G protein. So the G protein is in the off position. But when the G protein linked receptor binds the signal molecule, it causes the G protein to get rid of that guanosine diphosphate RNA nucleotide and trade it in for a guanosine triphosphate RNA nucleotide. And the GTP is an allosteric activator of the G protein. So the G protein has essentially switched itself onto the active on position. So we've gone from reception, that is binding the signal molecule, to activation of the first relay protein. Now we haven't got to activating the cellular response enzyme yet, but we at least got the first relay protein, the G protein, activated. And in studying these signal transduction pathways, what they found out is that there's 2 second relay proteins that the G protein can go to. There's a membrane protein called adenylyl cyclase which in some cells is relay protein number 2, that the G protein linked receptor, or that the G protein binds to and activates. And in other cells, there's a different membrane protein called phospholipase C. And anyway, in some cells, that is relay protein number 2. That's the relay protein that the activated G protein goes to and activates. So let's recap those two very briefly, and then we'll take it from there. If it's the cell type where the adenylyl cyclase is the relay protein number 2. There's adenylyl cyclase. It also is a membrane protein stuck in the membrane, but right now it's in the inactivated state because the G protein hasn't gotten to it yet. Here comes the G protein carrying its GTP activator. So now it activates adenylyl cyclase, and what adenylyl cyclase does is grab ATP molecules and make them into a cyclic version of that nucleotide called cyclic adenosine monophosphate. Here's normal old ATP. To change it into cyclic AMP, the 2 phosphates are removed, and that last phosphate, a new covalent bond forms that hooks it back onto the ribose sugar of the adenine nucleotide, and you get cyclic adenosine monophosphate. So this adenylyl cyclase, when it's activated, starts cranking out lots and lots and lots of that cyclic AMP molecule. And the cyclic AMP is not hydrophobic. It's hydrophilic, and it drifts down into the cytoplasm of the cell. And what it's actually going to do is this. The cyclic AMPs are allosteric activators of relay protein number 3, this one down here right there. And so eventually, just by sheer diffusion, one of those cyclic AMPs is going to end up drifting downward and activating that relay protein, like that. And then from here we get the kind of normal chain reaction that we expect from a signal transduction pathway. Now that relay protein is activated, it will activate the next relay protein, probably by kinasing it, that is phosphorylating it, then it activates the next one, probably also by adding phosphates to it, until eventually the cellular response enzyme is activated. And there's a term that we use for molecules like cyclic AMP that they are called second messengers. Second messengers are small, non-protein molecules like cyclic AMP that act as allosteric activators inside the cell for signal transduction pathways. Okay, now, so that was this part of the signal transduction pathway. Let's look at this type where the G protein activates phospholipase C. Here's the G protein, parked right next to the G protein linked receptor. The receptor binds the signal molecule that causes activation of the G protein, and now has a GTP activator. It goes sideways in the membrane looking for the phospholipase C relay protein. There it is. It's in its inactive state at this point. Here comes the G protein to activate it. All right, now what phospholipase C is going to do is it's going to start cranking out a second messenger called inositol triphosphate. Yes?
How fast do those processes go?
I have no idea. It's a good question. Like, in how many molecules per second would you get? I don't know. But, I mean, signal transduction can be very, very quick. Like, for instance, I know that some of the receptors on your tongue that allow you to detect sugars and things like that use G protein linked receptors. And you put the sugar in your mouth, and a fraction of a second later you can taste it in the sweetness in your brain. So it must be less than a second.
Like the adrenaline, like that, when it goes into your body, that's like instant too.
It feels instantaneous also. The adrenaline is actually slightly more complicated because certainly the adrenal gland is releasing that hormone into your bloodstream, and that's definitely taking place. But you also have nerve signals from your brain going directly to your muscles, and those also have an effect also. So part of what we can think of as your adrenaline rush is nerve signal more than the hormone, but both are certainly involved. Oh yeah, so anyway, so the phospholipase C starts making a second messenger called inositol triphosphate, or IP3 for short. And just like we saw with the cyclic AMP, it is a second messenger. It's small, non-protein, hydrophilic molecule, so it diffuses easily in the cytoplasm. But what is it? Well, the IP3 is actually a hydrophilic head group from some of the phospholipids of the membrane. In other words, what this phospholipase C is doing is lopping off the hydrophilic head from some of those membrane phospholipids. And there are different types of membrane phospholipids, but there's one type that has something called inositol triphosphate as part of the hydrophilic head group, and that's the head group that's released. All right, so phospholipase C makes tons and tons of this secondary messenger, the second messenger. And you might think at first that the IP3 is going to be like the cyclic AMP and that it's going to activate the next relay protein, but it's slightly more complicated than that. What happens is this: the IP3 drifts down throughout the cytoplasm, and it does not actually go to the next relay protein. Where it goes to is the endoplasmic reticulum. Let's see, where are you? There you go. This series of hollow membranous compartments near the nucleus. Inside the endoplasmic reticulum is a really high concentration of calcium ions. The little gray dots are calcium ions. So you get a large concentration of calcium ions inside the endoplasmic reticulum, but very few calcium ions out here in the cytoplasm. Well, what the IP3 does is it actually opens up a transport protein that's in the membrane of the endoplasmic reticulum. And when that opens up, just by diffusion, high concentration to low concentration, the calcium ions leave the endoplasmic reticulum and go into the cytoplasm. And it's actually one of those calcium ions that's the allosteric activator of the next relay protein. So now when that guy's activated, he goes to the next relay protein and activates him, goes to the next relay protein until you get to the cellular response enzyme. Sound all righty? Okay, now, if I can read your mind, you're probably thinking, my God, this is complicated. Why is this so bizarrely complicated? And believe me, biologists, when they were first trying to understand all this, were perplexed by it also. I mean, think about it. If you were wanting to make things simple, you could say, why doesn't the cellular response enzyme just get directly activated from the receptor? Wouldn't that make the whole world nicer if this receptor somehow just went right to the cellular response enzyme and turned it on? Why go through relay protein number 1 and relay protein number 2 and second messengers and yada, yada, yada to finally get to the cellular response enzyme? Well, I guess the shorter answer is nobody knows exactly why it's this way, because it's evolved millions, if not billions of years ago, and no one was around to watch it then. But that being said, biologists have come up with two pretty good theories as to why the signal transduction pathways are so long and complicated. And the first one has to do with allowing for different cells to respond differently to the same signal molecule. Let's get back to adrenaline for just a second, or epinephrine I should call it, that's its official name now. So this is this hormone that's released when you're angry or frightened. And when I first introduced it, I told you about what it does to the liver. Your adrenal gland releases the epinephrine into the bloodstream. Some of those epinephrine molecules go to the liver and cause it to release glucose into the blood to give you energy to fight or run away from whatever it is that's threatening you. But there are certainly other responses also. Some of the epinephrine goes to your lungs and causes them to get bigger, essentially, to give you more oxygen into your blood, to help you, I guess, run away or fight from whatever it is that is threatening you. And some of the epinephrine goes to your heart. It also has epinephrine receptors that causes your heart to start beating like crazy. And this is just the tip of the iceberg. The epinephrine actually goes to your digestive organs and does some changes there. I'd be surprised if there was really any organ in the body that didn't have a receptor for epinephrine. Anyway, so the point here is that it's the same signal molecule, but you get very different changes in the different cell types. The liver cell response is releasing glucose. Lung cells are relaxing to make the lungs bigger. Heart cells start working harder to contract faster. How is that possible? Well, this shows the signal transduction pathway for epinephrine. Its receptor is a G protein linked receptor. There's the receptor, there's the G protein, there's adenylyl cyclase. Here's some of the, phospholipase C. Here's some of the relay proteins. Here's the cellular response enzyme that will actually cut up the glycogen into the individual glucoses for the cellular response. This is supposed to be, I think, a heart cell.